entomologist. He works at berry crops and grapes, and he has been leading the charge in the state and also on national efforts for different spotted wing Drosophila research projects. And he's going to kind of give us an overview, a little bit about what's happening, what could be happening. And thanks a lot, Greg, for being here. I can't hear you, Greg.
Oh, how about that?
Beautiful.
There we go. Yep, I somehow pushed the button that knocked it off. All right, hi everyone. Good morning. So I'm going to share my screen, I assume, and see if I can find my PowerPoint. There we go. And let's see if I can get it into presenter mode. So as Laura said, I'm entomology with responsibilities for grapes and small fruit. And my lab is located at Cornell Agritech in Geneva. Before I get going to this, I just wanted to kind of make an announcement. I think for those of you who also have interest in tree fruits in general, I wanted to let you know that two things. One, you probably have heard that our tree fruit entomologist, Art Agnello, is retiring as of April 1st, and congratulations to him. We'll miss him. But in addition, we have been given permission to search for his replacement for a faculty line with tree fruit entomology responsibilities, and you'll be hearing more about that over the next few weeks. Advertisements and our call for applications is going out, I think, today, and we hope to be, processing applications and interviewing for candidates this spring, sometime in May. So we'll certainly want to be in communication with those people that have interest in tree fruits and have an interest in tree fruit entomology. Okay, so today I'm going to kind of focus in on kind of an update on spotted wing Drosophila. Believe it or not, it's been with us for 10 years. I don't know if any of you remember, and we've given obviously a lot of talks about it as we've learned more about the biology and how to manage this thing. And if you remember back to 2012, we knew it was coming, but even then we still saw a lot of damage and a lot of crop loss due to this invasive fruit fly. Over those 10 years, we've learned a lot about its biology and its management, and I think we're doing a lot better job. So I believe we've kind of moved from that crisis situation to something that is manageable, although perhaps not sustainable. We still heavily rely on insecticides for its control, and that's creating a number of problems, such as insecticide resistance, as well as costs and environmental impacts, things like that. So there's still challenges remaining with spotted wing Drosophila, even though we're doing much better than we were. And here's just kind of 4 of them, and I'm going to focus in on 2 for today. So as I said, insecticide resistance has always been a concern when we're needing to spray once a week in a crop and maybe have to put in 4 sprays on one crop. And then if you're doing a different crop, maybe some sprays in there like blueberries to fall raspberries. So insecticide resistance is a potential problem. And in fact, we are now seeing evidence of resistance out in California and a few other places to Entrust, our organic alternative, and a little bit maybe to pyrethroids. I'm not aware of any control failures, at least in the eastern United States, in any of our insecticides, but you got to figure that's coming. Fruit flies have a lot of generations per year, and you'd imagine selection is there for them to develop resistance. Another issue is the fact that, and I think we've talked about this before, is spotted wing Drosophila can use a lot of different hosts, both crops and non-crops, wild hosts. So that they're very abundant in the landscape. And they're really hard to reach for control in the landscape. You can't spray out there. It's not legal nor a good idea. And therefore, they're building up on these wild hosts and clearly moving into our crops. So that's an issue. We have, in general, lack of effective alternatives to insecticides. So there's a reason for relying on insecticides. We are making progress on that. We need to make more progress on being able to provide other approaches to management, cultural, biological, and behavioral approaches that can supplement insecticides or perhaps in some cases replace insecticides. That's one of our goals. And related to that, Unfortunately, when you have these invasive insects or other kinds of pests come into a new environment, they often don't bring their natural enemies with them, and your native natural enemies sometimes are not very effective. And that's definitely the case for spotted wing Drosophila. If we've looked at it, other people have looked at it, we're not getting much help from our native natural enemies. So the population's what we call a kind of predator escape, and you often can get very high populations like we see in spotted wing, where they're not getting much regulation from predators and parasites and parasitoids. So this area of lack of effective alternatives is something we've been very interested in, and as well as trying to do something about a better biological control. And I'm going to talk about those two things more today, but I'm happy to answer other questions. Before I get into these alternatives, I wanted to bring your attention that I'm part of a grant that just got funded by USDA, SDI, that's a specialty crops initiative grant, a CAP grant worth, I think, something like $6.5 million that several of us are involved with at Cornell, myself and Miguel Gomez in particular. This project, the PI for it is Ash Seal. Some of you might remember him. He was a postdoc for a short while in my lab. He's now at the University of Georgia. And then it involves a number of different states listed here on the slide. The idea is that, as I mentioned before, we're moving from this crisis response. Now we want to develop sort of more sustainable long-term management solutions, best management practices. These are the project goals. On the next slide, I'm involved with most of these. The first one is to develop teams in different regions where we work with specific growers, 3 to 5 growers, and work with them to implement what we have right now, our best management practice. And we plan to be doing that in New York, and I'll be involved with that with several of you. And I am going to do this work actually probably mostly in blueberries. Second objective that Miguel is actually heading up is developing decision aids, economic models and tools that growers can use to manage spotted wing in a most profitable way. The third objective is developing some of these alternatives, both biological control and behavioral control. And I'll talk a little bit more about that in a second. There's an objective on this insecticide resistance problem and developing resistance management plans. And finally, an objective just in terms of communication and helping growers basically and optimize and make the best decisions possible. So that's happened. It just got funded and will be funded for several years. So that's good news, and I think we can continue to make some progress there. So let me kind of go back to those two kind of different alternatives. The first one I want to talk about, which I'm not going to spend a lot of time with because we have talked a lot about it, is a cultural control technique related to excluding these flies from the crop, exclusion netting. And this is something that Delilah Riggs, many of you know Delilah, and she's been a real driving force behind this. Is something she heard about, and she really developed it on her farm. And I'm going to show you a slide, and it's worked quite well for her. We've helped kind of monitor infestations at her farm, comparing it to some other farms over many years, since 2014 actually, right into 2020. We've done different things out there, but the basic idea is how well has she been able to manage spotted wing infestations without using insecticides within these netted areas. And these are all the different years here. I don't expect you to look into it, but basically the netting has done better or as well as chemical control every year. We've kept the damage quite low within the netting area. Where we've sometimes seen some increases is right at the end of the season, actually beyond harvest, we'll get some flies in there. And that is a problem with netting. As you get flies in there, they can be successful. And that's an area we need to keep working on. But in general, over a 6-year period, it's been quite effective. So, and I know you've heard a lot about that. So now Delilah developed something that worked well for her. But it's kind of unique to her situation and what she had available. And so we got really interested in saying, okay, this can work. Can we develop a process for the support systems for the netting that growers could use or companies could use to put in for growers to make this thing a little more usable for growers if they want to adopt this kind of approach to blueberries or other susceptible crops? And so we put together a project, and one of the things that, I'm an entomologist, I'm not an engineer, so we kind of played around for this for a couple of years, but really didn't make much progress. But then Delilah got in contact with a colleague of ours at University of Vermont, Chris Callahan. And Chris is an agricultural engineer with an extension responsibility. Very great guy, and he's really brought this project along because he brings that engineering background. How can you develop these support systems cost effective, but that last and it can hold up to various kinds of situations. So Chris came aboard. Laura's been involved with this project. Esther out in Western New York, myself, and we've been working cooperatively, obviously, with Delilah and Technic Industries, who produces the netting that we have shown can be effective, and also a company called Gintech Shade Technologies that was building support systems for ginseng, and they've been adapting that to things like blueberry crops. So this project is really a demonstration project, and we started in Eastern New York at Delilah's farm with this post and wire system, and that's what I'm going to show you a couple pictures of. The objectives of this project were to really develop and implement engineered plans for this support system, bring that information to growers on how to construct these kinds of support systems, how they could do it, develop a materials list and a time budget so we can get an estimate of the cost. And then I think importantly, we would love to get NRCS involved or to accept an application to have netting and support systems for netting as some sort of cost share program, because it certainly can greatly reduce the amount of insecticides being used on farms if implemented. So that's kind of our longer term goals. I'm just going to show you a few pictures. I said we set this first demonstration up this last spring and summer, and it's amazing we actually pulled it off in the era of COVID. We put in the proper precautions. Luckily, we're working outdoors, so that makes it a little easier as well. Many of us worked on this. This is just a picture of the metal post system and some of the support wires going in. This is putting some netting, pulling the netting actually over the sides of the blueberry planting across the support system. This is when it's almost complete. So I just wanted to give you a feel for what that we got that up before July and got a full season of it. You can see it's a very open, roomy environment underside there with plenty of space for the plants and people to work. It's pretty comfortable in there. So I think overall that was a good first experience with this system. We based it on ginseng's kind of design for supporting coverings on various things, and it worked out pretty well. Now, Chris had documented the amount of time we've spent, this material cost, etc., and has put together an estimate initially of costs. And you don't need to, you can kind of look at this, and we're going to post this later as a PDF so you can look at it more carefully. But it's not the installment and getting it set up is obviously not cheap. For steel, for a per acre was about $28,000, a little over $28,000. We actually liked the steel support a little bit better than the wood, but both of them were successful. You can see the cost where it comes from materials versus labor. The netting is supposed to last quite a few years, I mean, over 10 years, especially if you don't have places where it's abrading. And the steel support, I think, will last quite a bit longer. So it's an initial investment, but it should be around for quite a while. And it not only provides some protection from spotted wing Drosophila, but also from hail, keeps birds out, that kind of thing. And I can answer questions at the end. I better move on with my time here. But I just want to mention, we are setting up something similar to Western New York this year, that we'll hope to have events. We will have events. Hopefully we can do it in person rather than over Zoom, where growers are interested, people can come out and actually see the product. We'll provide information on its construction, etc. So that's something to look forward to, hopefully this summer. Let me switch gears now and talk a little bit our work and other people's work on this approach to management based on manipulating the behavior of these flies so that they don't infest your fruit. And I have this kind of diagram, I've used this before, which illustrates the concept of push and pull, where if you can design something that the flies don't like or makes them so they don't recognize the fruit, a repellent, for example, to keep them from colonizing a planting, and then have something outside or within the environment that's very attractive to them that you put a kill agent in, and I've just got a couple of examples here. The idea is that you can manipulate the behavior, kill them in these attract and kill traps, and keep them from, or reduce their chance of infesting fruit. And we've been working on the repellent side of this question. Well, we've been working on both, but we've really been focused on repellents. Our dream would be find something like in the biting fly world, like DEET. DEET is incredibly effective. And can we find something that the flies really don't like that would be similar to DEET? That would be our kind of holy grail. And we haven't gotten there yet, but that's what we'd like to find. Both something that they don't like and also that you can spray on plants that doesn't affect people and it doesn't affect quality and taste and that kind of stuff. So we looked at 1-octantriol, the mushroom alcohol. It has good efficacy against the flies, but it has a pretty bad odor. So we kind of dropped back on that. Geosmin's another chemical that's found in soil bacteria. Flies don't like it, at least in the lab, but over longer term, that seems to break down, so we've abandoned that. We've been looking at the odors of Botrytis cinerea, the causal agent of gray mold, and that looks promising in that the flies don't like the odors from Botrytis, but we haven't figured out what the actual chemicals are yet, so that's a project ongoing. This last one on the bottom is 2-Pentafuron. This is a chemical, this compound we accidentally discovered looking for attractants. It is it's a food additive. It's approved as a food additive. It has a mild odor. So it's something that we wanted to pursue a bit more. It showed good activity in the lab in repelling or preventing the flies from finding fruit. We've tested it in the field at a couple different scales. This is just the data for the small inflorescent scale where we had these little plastic packages that was releasing 2-Pentafuron out into the area around this fruit. And then we measured infestation over a certain time period. And we see a significant reduction in infestation. It's not zero, but we dropped it by about 60%. And that's kind of what we've been seeing on these small scale experiments. Okay, well, that's a small scale. Can we get this to work a little larger scale? So this past summer, we took advantage of these micro plots of raspberries that we have at Cornell Agritech, and we put out a bunch of these plastic releasers at some plots and a little bit fewer in other plots, and then measured infestation in the plot. And this did not work as well as we had hoped. This is the results. And the only place where we saw about a 30% reduction was at the highest release rate. I think we had 32 of these little plastic sachets out there. We don't know exactly what the issue is. We think we just didn't create a high enough level of the compound to give us the protection we wanted. And we're looking into other ways of releasing it. These sachet or these plastic sachets is probably a limitation at this scale as we scale up, and we're working with a company to try to develop a different way to release this. But it also raises the possibility that maybe we need to combine our spatial repellent with something else. I mentioned DEET, and really the interesting thing about DEET is it works on different levels. It works as an odor. It works at the level of odors, and it messes up with mosquitoes' odor reception. It also messes up when they taste it. They don't like it. They have gustatory receptors, and there's things that they can detect with their feet that they don't like. So it's working at different levels. So can we find something that maybe works as a contact against spotted wing that we can combine with a spatial repellent? So we started working with neem or azadirachtin. And we can show in the lab that if you put neem on a raspberry, you can reduce infestation at the sort of 4 times the label rate that we use in the field. Can we combine it with 2-PF and kind of get an additive or a synergistic effect? That was what we were looking for, contact and spatial. And we tried that out this past summer. We're going to look at it again, and it wasn't totally successful. D represents our 2-PF. So we got a good effect of 2-PF. We got some effect of azadirachtin, A, reducing it by about half under this field experiment. But the combination didn't do any better. In fact, it was basically almost the control, or all the control was really coming from 2-PF. So, but this is an area we're looking at where we're working now with another contact oviposition deterrent that looks really promising. I can't really even reveal any more than that about it because it's going to be patented. We're working towards patenting it, but if we can get it in a place that we can apply in an economic way, I think it has some potential either with 2PF or without it. I need to keep moving along here. We're also working with our colleagues on some other approaches to attract and kill. This is just a graph showing that we get field efficacy with this product that's still in development stage called Splat Hook. And this is this wax matrix that you put on around the planting or in the planting that includes an attractant for spotted wing, a food stimulant or a phagostimulant so that they eat it, and a poison, a toxin, and it kills them. And it's shown some efficacy in the field. It also has not worked in some situations. So it's something that we're interested in working at more and seeing maybe if we can combine it with our repellents. Another product developed by a colleague of mine out in Oregon, Von Walton, is this food grade gum. He's now formed a company that under the right circumstances has shown pretty good efficacy in the field or semi-field conditions. And this is just a bunch of data comparing infestations untreated versus gum. What the gum is, it's some sort of attractant or arrestant, and they can put a pesticide in with it, but the flies get on it and they just kind of hang out there. It needs to be kept moist, so it has some limitations there. But again, it's something that, although not 100% effective, has some promise. And there's some other things going on with attract and kill that I think is worth looking at. So over the next year or a couple of years, that's kind of where our research is going on this topic. We want to get to this place where we can, in a cost-effective manner, come up with something that manipulates their behavior. Maybe it won't be the answer to the whole season, but if we can just get something to work for several sprays, we can reduce that pressure on insecticide resistance, allow natural enemies, and I'll talk about that in a second, to be more effective. So that's kind of a future goal. Finally, I wanted to mention the work on biological control. As I said, the native natural enemies aren't doing very well. So our colleagues at California at USDA have been exploring going back to Asia where spotted wing comes from and looking at what controls them there. And there are a number of parasitoids, these tiny wasps that lay their eggs in the larvae of spotted wing. And we have some of these types of organisms here, but the ones we have in North America are not very good at killing spotted wing, but the ones from Asia are. And my colleague Kent Dana, who's kind of taking the lead on this, has come up with 3 species, and in particular this one species on the upper left corner, Gannapsis Braziliensis. Looks quite promising. It's quite selective to spotted wing. It doesn't seem to attack many other kinds of fruit flies, and that's the one we're pursuing a permit to get a release into North America. I mean, that's quite a process. It's a 2 to 4 year process to get a permission to release an insect. There's a lot of precautions you have to to take before you release something foreign into our environment. We're very close down at the bottom here. We've now received the tribal permission, and we're now working on getting public comment. And right now, Kent expects a positive outcome for this permit by the end of this summer. The plan is for a couple of us on the Northeast, myself, Phil Fanning up in Maine, and also Cesar Rodriguez Sauna, to get colonies started in our labs, and then start doing some releases in the Northeast to see if we can get it established. And if we can, remember I talked about that wild host, what can we do about those population of wild hosts? This is what I'm thinking about. If we can get these parasitoids established, they should provide some control on these wild hosts, reducing the overall kind of pressure from the landscape, which should help us in terms of management. Those are a lot of shoulds. It's going to take a few years to see if this thing can be established here, but that's where we're heading. Okay, I want to stop here. I haven't been a chance to look at the chat room, but I just wanted to thank you. And if I have any time for questions now or later, I'd be happy to take them.
Thank you very much, Greg. I think people are just trying to concentrate on what you're saying and didn't chat into the chat box yet.
Okay.
And how we've been trying to do this during these workshops, if the speakers are able to stick with us for the whole hour and a half of the workshop, please just put your chat question in as it comes to you. Even if somebody else is speaking, we can direct the chat, those questions later to the speaker. But I have one. I'm wondering if you're going to try to go down the route of using those chemical behavioral attractants and repellent methods. Do they have to be used singly, or can you combine them with insecticides to create a better, more robust management scheme?
Yeah, I think that's probably true. And there's a bunch of different ways we could do that. To make it cost effective, you'd hope you could use it, maybe behavioral control the first few weeks when spotted wing's out there. And I think this could really work in blueberries. You might get through your harvest with it. Fall raspberries, probably a more challenge, and then maybe you have to put one spray on. I mean, you could put one spray on top of it as well. I think something interesting is being developed that is kind of this idea of a phagostimulant, a feeding stimulant that with an attractant that you mix with your insecticide. There's a new company in Europe that's developing a product. It's not labeled yet. Some of our team is going to look at it in research mode this year, but perhaps by next year, we might have something that we can test in blueberries. The idea is you mix it with whatever insecticide you're using. You use a lot less water. They recommend 95% reduction, which seems crazy to me, but some reduction, and the droplets have sort of this attractant in them that's supposed to really boost the efficacy. And they're trying to get it labeled for a sale. It can make 2 other insecticides that you, the grower, producer, mixes. So that's kind of what you're talking about. And I hope it works. The companies, companies are always going to promote it. But that'll be an easy one for us to implement, and it would at least save spraying time, maybe save number of sprays, that kind of thing. We still have the resistance issue, but yeah.
Yeah, great. Yeah, it would be just really nice to be able to reduce the numbers of sprays. That would be a huge, huge benefit. Well, thank you. That was really interesting.
You're welcome.
I appreciate you. And so now I guess if I don't see any chat in the chat box, we'll just move to our next speaker. And Lynn Szysnoski is here. Lynn has been with Cornell for a little over a year, maybe, Lynn? I think it's, but Lynn, it came directly from California, although she does have a lot of experience with weed systems throughout the country. She's been in a couple different environmental locations in the country. So her switch to the Northeast is a really good move for us because she brings a lot of experience. And Lynn, I don't know if you have a camera, and that's okay if you don't, but if you do, you can go ahead and turn that on, or you can keep it off, whatever you feel more comfortable with.
Well, I would put it on, but my husband and I both are using the internet and we have limited bandwidth. So
okay.
I'll keep it off,
Great.
or you will lose me entirely.
Okay, no problem. So Lynn is going to talk about making sense of herbicides and herbicide activity. And thank you so much, Lynn, for being here.
Yeah, so this is going to be, this is actually going to be sort of a background talk of sorts. Not saying use this herbicide against this weed at this rate, at this timing, but rather just kind of go back to the basics and how do these products work and how are they damaging plants and what kind of symptomology do they show when injury shows up, just to get help get some foundational information about choosing herbicides in systems and understanding injury potential. So blueberries are sensitive to weed competition, open soil, lots of light, and that facilitates the development of weeds as much as crops. And so there is a lot of reliance on chemical weed control for the management of unwanted vegetation in these systems, and that's herbicides. And herbicides are just chemical substances that are used to control unwanted plants. And in the grand scheme of things, with respect to weed management, herbicides are really one of our newer technologies. We've been physically controlling weeds for a long, long time. But the chemical era of weed control that we know right now, that are synthetic herbicides that started with, the advent of, 2,4-D, is very new. Prior to the 1940s, we did use chemicals. They were inorganic substances like sodium chloride for control of field bindweed, but we were using them at obscene rates, at tons per acre. Orchard heating oil to control wild onions or lime for horsetail species. But, so this is our newest technologies, and really some of our most optimized and for many systems cost-effective technologies. Now, we talk about herbicides and we classify herbicides a lot of different ways, and that can get confusing because sometimes we tend to use these words interchangeably, and sometimes we combine them together and we use them in some instances and not others. So we classify herbicides in different ways by where they're applied or when they're applied. So soil versus foliar, prior to weed emergence, following weed emergence, how they move in the plant. So for our foliar products, are they contact? Do they just control tissue they come into contact with, or are they systemic? Do they move through the plant to sites of action where they then, cause damage? What types of weeds do they control and how many? So do they control a lot of species or fewer? Are they selective, such as broadleaves only or grasses only, or are they non-selective? They'll get all species. And then we classify them based on their WSSA groups, which refers to specific cellular targets that the herbicide active ingredients work on. And that's when we're talking herbicide resistance management. We talk about rotating your WSSA groups or rotating your sites of action. That's what we're referring to, those biochemical activities within the plant and those targets. And then how they kill the plant in general, this greater mode of action. Are they just inhibiting, growth and development? Or are they, generating reactive oxygen species? And we combine these terms. So glyphosate, that's the active ingredient in Roundup and other products, is a foliar applied post-emergence systemic broad spectrum non-selective herbicide. Clopyralid, which is in Stinger, is a foliar applied post-emergent systemic, but it's narrow spectrum or a selective herbicide because it's only active against certain families of broadleaf weed species. Now, if we look at all of our herbicide categories, our WSSA groups, there's a lot of them, but they're changing. We've recently had some drop off. So the WSSA 7, 8, 11, and 16, those groups don't exist anymore. And what was in WSSA 7 is now wrapped up into WSSA 5. But then other WSSA groups have come along, WSSA 18, 23, 24, et cetera, et cetera. So just to let you know that this is, there are changes that are occurring and that these categories aren't necessarily static. But what we really do want to focus on is, again, they are acting within the cells themselves. These herbicides are interrupting biochemical processes, and that's what's killing the plant. If you are absolutely curious about changing herbicide classifications, you can go to the HRAC global site, and they will have what the new categories are going to be, and they're going to have a giant map that's going to have all of the herbicide active ingredients on it and their groupings. So again, herbicides are chemicals that act at the cellular level in plants to cause whole plant damage and death. And so what are those groups that are in blueberries that we're going to be familiar with, and how are they working? So number one is our grass herbicides, those ACCase inhibitors. That's going to be clethodim, sethoxydim, fluazepop. What they do is they interfere with fatty acid synthesis, and fatty acids are components of our cell membranes. And so if you can't build cell membranes, you can't have actively growing plants. And that is how these herbicides kill grass species. Note, they're not going to get grass lookalikes. So they don't have any activity against nut sedges. So these are systemic products, and you know what? You have to apply them to actively growing grasses. If you have grasses that are heat stressed, that are drought stressed, they are not going to respond well because these are systemic herbicides. And they, they need to be moved within the plant, and they need active biochemical processes that are going on. With respect to injury, the damage you're going to see from the lipid synthesis inhibitors is going to be yellowing and wilting. you could see reddening and purpling, particularly of that newest tissue. And then what's going to happen is that new tissue is actually just going to die out. That center of the grass is going to die out. And we have this thing called dead heart, where it just dies. And then this is a corn example, and it just falls out. And this is another picture of corn treated with that got treated with a grass herbicide. You can see that yellowing, you can see that purpling, and that centered dead tissue. The next category is our acetolactate synthase inhibitors. And what they do is they interfere with the synthesis of a specific set of amino acids, the branched chain amino acids. And amino acids are building blocks for proteins and other plant structures. And if you can't build proteins, you can't carry out enzymatic activity. If you can't build plant structures, you can't grow and develop. And that's how these herbicides work. They're stopping growth and development. They can have pre and post-emergence activity. The post-emergence is usually on very small species. They're not very effective as plants start getting larger. But one good example of post-emergence activity is actually on yellow nutsedge, where we recommend sequential post-emergence applications at the 3 to 5 leaf stage. Note that with the pre and the post-emergence activity, that some species that are controlled pre-emergence are not necessarily controlled post-emergence, and vice versa. So you have to be very careful how you're using it and what you're targeting. So it can be most effective. And again, we talked, the active ingredient is halosulfuron. Injury in response to amino acid inhibitors is wilting, and you often see red or purple veination. So you get, a dark purple coloring in the veins. You might see the undersides of leaves turning purple. And then you can also see chlorosis and leaf deformations, and sometimes very, 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 very low rates of the ALS inhibitors can actually cause symptoms that look sometimes like 2,4-D or dicamba or clopyrrolate or some other auxin damage to the plant. So very low doses can actually be a little bit confusing. So that's why it helps if you also have that purpling show up, because that helps maybe target it for being more like amino acid damage. So this is actually an ALS inhibitor injury on hemp. And you can start to see some of those newer leaves are starting to get a little strapped. They're getting a little bit longer and thinner and a little twisted. ALS injury, but might start to look like a little bit of auxin injury. The WSSA3 are our microtubule assembly inhibitors. This is going to be pronamide, pendimethalin, oryzalin, or yellow herbicides. And what they do is they inhibit cell division. They actively interfere with the development of the cytoskeleton within the cells of the plant. And so if these cytoskeletons don't develop, and cells can't divide, that means that the plant can't grow. So it inhibits growth and development. These tend to be grass herbicides, but they do get some broadleaves. So the injury that we see on our seedling growth inhibitors, swollen stems. Pendimethalin is a good one for this. You might see plants that start to get really swollen right there at the soil line. Stubby roots. The roots don't completely develop, so you get these short little, kind of witch's broom, fat and thick, stubby tissue. And even though these are soil applied herbicides, if they get onto leaf tissue, they can cause damage. So they show up as kind of puckering and cupping and other leaf distortions, and can result in plant stunting. The WSSA4 category is our synthetic auxins. So these are mimics. They're growth regulators, and auxins don't really have specific targets. One unique target in the cell. They go after a lot of different processes that are responsible for plant growth and development. So cell division, they interfere with cell wall plasticity, protein synthesis. They interfere with they can stimulate ethylene, which can induce stress response pathways in the plant. So that has effects on cell division and cell growth and cell wall plasticity. That's why you see the curving and the twisting and the bending, because they're interfering with that, and the cells are growing in different directions, or they're not growing equally on all sides of the plant, and that's causing the symptomology that we see. Not all auxins are created equal, and I got some calls about working on one species, not another. So like 2,4-D and clopyralid are rated for good control on something like horseweed, but for a nightshade, clopyralid is fair at best, while 2,4-D has a good rating. So they're not all going to be created equal, and weeds aren't going to respond equally. So this is that twisting and that epinasty and those distortions that we've talked about, crinkling, puckling, leaves becoming really long and thin. That's what we call a strapping sort of behavior. Here's some more symptoms. The left is that's hemp, so twisting and bending and leaf deformations. The middle is a cupping. And then the rightmost, obviously, stem epinasty and twisting. So really good symptoms of what auxin herbicide injury is going to look like on plants, crops and weeds alike. So the next group is the Photosystem Two Inhibitors. This is going to be hexacinone, terbicyl, simazine. They're all in different chemical groups within this larger WSSA5 group. So what happens with these chemicals is they inhibit photosynthesis. They bind to one specific protein that's involved in the photosynthetic pathway, and it stops carbon dioxide fixation and the production of chemical energy that's required for plant growth. But death isn't actually due to the plant not having nutrients or the plant not having energy. It's not like it just shuts down. What happens is we have a buildup of reactive molecules, and those reactive molecules, those reactive oxygen species, just go around the plant and just destroy proteins and lipids. And that's why these herbicides have burning activity. So they can get some annual grasses depending on the herbicide. They're good against annual broadleaves, and then even some perennial species, again, depending on the herbicide. So the injury that we see with the Photosystem Two Inhibitors, it often starts off as like a water soaking on the edges of the leaves, which then becomes a necrosis. And you see a burn back from the very edges all the way back to the base of the leaf. Between the veins, that area, part of the leaf tissue, becomes chlorotic, and those veins will still remain dark green, and that's a symptomology of the Photosystem Two Inhibitors. And this is some pictures, and I'm using hemp because I'm also doing work in hemp. Hemp is sensitive to so many herbicides, and it's actually really great for getting beautiful pictures of herbicide injury. But this is a Photosystem 2 injury on hemp. And you can see the edges of the leaves getting water soaked. You can see the necrotic tissue burning from the edges of the leaves back to the bases. And you can see those dark green veins. And you can see that yellowing and that loss of pigmentation in between the veins. Number 9 is glyphosate. It's 5-enolpyruvylshikimate 3-phosphate synthase inhibitor. It's glyphosate. It's the only herbicide in WSSA9. This is also an amino acid inhibitor like the ALS inhibitors, but it actually interferes with the synthesis of a different set of amino acids. It's the aromatic amino acids, so tryptophan, tyrosine, phenylalanine. But again, it interferes with the development of proteins, of enzymes, of plant tissues. These are non-selective post-emergent systemic herbicides. And this is one here in the state and around the US and around the world we're very concerned about, because in the 1990s, we started developing herbicide resistance. And with the advent of glyphosate resistant crops, we now kind of have seen kind of an explosion of glyphosate resistant weeds, and that includes horseweed here in the state of New York. I just did a resistance screen of 30 populations, including some from Eastern New York, and 90% of our populations are resistant to Roundup. So we need to really be thinking about, we still have a lot of utility left, obviously, in this herbicide, but we have to pay attention to resistance development and the species that are susceptible to that concern. So injury due to Roundup, witch's brooming, basically shortened internodes. So the internodes between leaves don't expand, and so they all look kind of clustered, like you see on the leftmost picture. Chlorosis that starts at the base of leaves, and then low doses of Roundup, very much like the ALS inhibitors, can be mistaken sometimes for auxin injury on many plants. So this is some of that symptomology, again, just showing that chlorosis, it's on the newest tissue because this is a systemic herbicide that's translocated in the phloem to meristems. But again, you really, one of those distinctive characteristics is you start to see that yellowing that starts at the base of the leaf, right where that petiole, joins the blade. That's where we see that yellowing start to develop before we start to see, full chlorosis and then necrosis and plant death. WSSA-10 is a glutamine synthase inhibitor. This is glufosinate. Again, the only herbicide we've got in this group. What it does is it inhibits the enzyme that converts ammonia to glutamine and ultimately inhibits nitrogen metabolism in the plant. But really, the damage comes due to the fact that we have a buildup of ammonia within the plant, and that ammonia is really damaging to photosystem 1 and photosystem 2. So that's why we kind of see some burning symptomology associated with glufosinate activity. This is a contact herbicide. It does get grasses and broadleaves. There is some limited movement in the plant. So it's not as fast a burn down sometimes as some of our other contact herbicides like carfentrazone, which is AIM, or paraquat. But it's also not as effective, and as systemic as something like glyphosate or clopyralid, for managing plants that might be perennial. Okay, so again, symptomology, there is chlorosis and necrosis. So you'll see spotting on tissue if you would have glufosinate drift. And burning from this, nitrogen buildup that burns the tissue away. The WSSA-14 category, these are our Ppo inhibitors. This is going to be Chateau, flumioxazin, and carfentrazone, which is the herbicide AIM. What it does is that it inhibits the development of chlorophyll, but the plants don't die because they're not making they're not photosynthesizing properly. Really, what happens is that we get those reactive molecules building up again. Those reactive molecules move around the plant, or those reactive molecules move around those cells that they're in contact with, and it just destroys plant cell membranes and just breaks these plants down. They can have pre-emergent or post-emergent activity depending on the active ingredient. Flumioxazin is mostly a pre-emergent herbicide, but has some post-emergent activity on very small weeds, whereas carfentrazone is a post-emergence only chemical. Primarily active against broadleaf weed species. Looking at some other herbicides that are also going to have activity against grasses, cell membrane disruptors. They're going to look, I'm going to not lie, kind of close to what we're going to see with glufosinate activity, chlorosis, necrosis. We might see some leaf distortions, but a drift event is going to look like necrotic spots. By the way, with some of these Ppo inhibitors, like the soil applied Ppo inhibitors, dust that's treated can get kicked up during like mowing activities, and that dust can come into contact with leaf tissue and can cause burning, splashing, sometimes can splash, water splashing can splash the herbicide up and onto sensitive tissue. Cell membrane disruptors, again, you see that burning of that tissue, it comes into contact. When I was talking, the splashing, that center one, that's cotton. And that's actually injury due to a Ppo inhibitor in cotton. There's strict guidelines about how to use it. What happened is that they got a rain event, it splashed up, it got on those cotyledons and just completely damaged them, but not translocated, so that new tissue growth is looking okay. The WSSA-12 category, this is going to be norflorazone. Again, interfering with the production of carotenoids that are pigments, associated with photosynthesis. Lack of carotenoids, again, reactive molecules build up, and that leads to the destruction of proteins and lipids within the plant. And I actually don't have any good pictures right now of the WSSA-12. The WSSA-27 category, this is our Hppd inhibitors. This is mesotrione. Again, carotenoid biosynthesis inhibitors. Again, ultimately, what happens is it's reactive oxygen species building up and getting burning of the tissue. But this category is actually going to have some unique herbicide injury showing up. Because it does inhibit pigments. We actually, with the Hppd inhibitors, with mesotrione, we see bleaching of plants that are treated or are accidentally treated or come into carryover contact with this herbicide. And we see whitening and translucid bleached foliage and chlorosis of the veins. And I want to bring this up because you might be looking at plants, you might say, oh, look at, okay, we're seeing, differences between the veins and the pigment inhibitors. So this group, the Hppd inhibitors, the veins become chlorotic and the area between the veins stays green. Remember we talked about the Photosystem Two Inhibitors? The veins stay green, but that area in between the veins, that's the part that goes chlorotic. So, it's kind of that flipped image, which is good for, determining, diagnosing, injury that you might be seeing. So the WSSA 15 and the WSSA 29, the 15, these are very long chain fatty acid inhibitors that interfere with the production of the plant cuticle, and that causes cupping and puckering and other leaf distortions. They're soil applied herbicides, mostly grasses, but with some broadleaf activity. They're on napropamide, which is Devronol and S-metolachlor. By the way, napropamide is one of those herbicides that got recategorized. It technically isn't a very long chain fatty acid inhibitor, but I'm considering it because that's what it was. It's been listed as, and labels haven't changed yet, but now it's shifted to uncharacterized, but it causes some of the same injury. The 29 group is diclobinil. It's also Allion and Dazaflam. Again, interfering with cell wall composition and structure, has very similar symptoms in damage to the very long chain fatty acid inhibitors, cupping, puckering, leaf deformations that are associated with them. And then this is the last category that we're going to talk about, the WSSA 22, which is a photosystem electron diverter. This is paraquat. Reactive molecules, again, build up and burn away tissue. And one thing for these molecules that burn away tissue, whether we're talking paraquat or the Ppo inhibitors or some of the other, like glufosinate, oftentimes I get a call and someone's saying, what is this? Is this a plant disease, or is this herbicide injury? And one good thing with these products is that their injury when they're kind of drift events tend to show up as circles. And these circles might be able to burn away like a vein, and they're not constricted by veins in the plant. So if you see, you know, damage and it's between veins and it's very angular, it might be a pathogen that, you know, is restricted by the veins and can't move out. Whereas a paraquat, that damage is going to occur across all of the plant tissue. So it can be a good diagnostic symptom for understanding, you know, if you're dealing with a pathogen versus a herbicide, particularly when you have a lot of little spotting like this. You know, if it's going over veins, it's probably a herbicide and not a pathogen. So I know I'm out of time. There's just some general notes about pre-herbicides and making sure you use them properly. I don't want to take away from the next speaker. One thing I'm going to say about the pre-emergence herbicides, these are soil applied herbicides. So be mindful of your soil type, and that is going to affect whether you can use a product, what rate you can use a product, and be mindful of what's on your soil. If you have a lot of leaf litter or other trash or anything that's going to interfere with the herbicides coming into contact with that soil, you might see really reduced activity. So you've got to be mindful that you need clean spray surfaces to really use a lot of the residual herbicides effectively, and you need them to be incorporated. So, you know, we need dry soils, don't work with residual herbicides. They need to be in the soil water solution, because that's where they're being taken up by those emerging seedlings. With the post-emergence herbicides, you know, whether we're talking, you know, contact or systemic, the sooner that we can put these herbicides down, the smaller the plant, the better our control. And just pay attention to, you know, your spray volumes and your water quality. A herbicide like paraquat, you need a big spray volume because you need to cover as much plant tissue as possible. Something like glyphosate, lower spray volumes because you want that herbicide concentrated in the spray droplets for a better efficacy. And thanks for your time.
Thank you, Lynn, very much. That was a really good review. And I just wanted to, there was a couple of chats in a couple of questions. One from a grower that's wondering if there's any thoughts or recommendations, hesitancies concerning any of the OMRI approved burn down products in conjunction with mechanical weed control.
Yeah, so that's actually an excellent question. And that is probably not something that we've investigated. Are we talking like kicking up the dust and maybe causing injury, or just, you know, treating a plant and then cultivating and then having reduced efficacy?
I would say the second might be the more likely scenario, but I don't know if Kevin, if you want to weigh in, you could even unmute yourself if you'd like to.
So you know, a lot of our OMRI products, they are contact herbicides, they are burners, right? You should start to see activity right away. So I think if you're, you know, you're going to go in, what's going to happen is they don't have any residual activity, obviously, in the soils. They don't have systemic activity. You know, if you burn them and you cultivate them, probably what's going to happen with your cultivation is you might stimulate new weed seed germination, and that's going to come up, and then you're going to have to go in again and manage it some other way, whether it's another burn down. They're expensive and they need giant spray volumes. So, you know, obviously be mindful of that. You know, for broadcast, you know, those applications over a wide area, you have to have high rates, lots of water.
Yeah, that's great. And I think the final notes you made about the suggestions about pre and post and ways to make sure you're getting the best efficacy, those are really important. And they might actually have, there was one question about kind of an overall plan for, you know, a annual plan for herbicides. And I think we rely on just a few chemicals, at least in my you know, my experience with blueberry growers, that pre-plant, using pre-emergence there in trying to do that effectively is really important. And we might be able to have an entire talk just on that, actually.
So I will probably write up those last 2 slides at some point in time in a newsletter article that you can put out, Laura, about understanding the factors that influence residual herbicides and post-emergence herbicides and to maximize them. Also, where we're going to have issues with residual or with resistance developing, pre-emergence herbicides put less pressure on our post-emergence herbicides for resistance development. If we have a lot of perennial weeds, our pre-emergence herbicides, with the exception of Casseron and maybe a few others against very specific species, aren't effective. So that's another.
Right. That's another problem. Yep. There's one question here that I think I got privately. Is there a new type of 2,4-D in blueberries as a spot spray? Are you aware of that?
Yes. So I did see something out of New Jersey, and I'm going to need to check that for New York, the 2,4-D choline. So 2,4-D choline is a low volatility or formulation of 2,4-D. It's marketed as a low volatility formulation of 2,4-D, and I think it is registered, but I have to look. Like I said, I just saw something come out of New Jersey about the 2,4-D. Now, low volatility does not mean no volatility, but it is also registered for use in Enlist soybean systems. So we do know we've got that product in New York.
All right, great. Thanks, Lynn, very much. I'm gonna ask Natasha to put the polls out there, and these polls are really important for those of you who want DEC credits. There's 3 questions, and we want you to answer the 3 questions, and Natasha will kind of keep them. And I did actually, before Esther talks, I just wanted to pester Lynn again. Would you mind, Lynn, just kind of describing the research you're going to be doing in mechanical cultivation and some of those cultural controls of weeds? Would you just say a couple quick words about them?
Yeah, so most of my focus, I've only been here a year, so my focus has been in the berries, or not berries, in vegetables. I am starting some new projects in grapes and tree fruit systems, so perennial crop systems would include berries. I have not looked at the mechanical, like the cultivation yet. We do have a, knock on wood, project in to look at an electrical weeder that looks like a mower that you can go right up close next to the plants. It's gonna that Laura will be involved in, and again, knock on wood, that we're hoping we're getting, that we think could be a really effective strategy for managing weeds, because we're not putting chemical outputs into the system. It might have some activity against perennials, so we're really interested in that. I do have a new vision guided sprayer that's an automated sprayer that I'm going to be looking at in perennial systems to can we start reduce our overall post-emergence herbicide use by spot spraying, because it sees a weed and it sprays just the weed, and you're not spraying any kind of bare ground area with a post-emergence herbicide where it's not doing any good. And we're going to be looking at that in perennial crops. And, is that going to be a tool for maybe bringing, using some more expensive chemicals, like say maybe the organic herbicides, if it can just see the weed and it can just spray it, and reduce the giant volumes that we need to put out. Or can we bring new herbicides online for use that a plant might, the berries might be more sensitive to, but now because we're not putting it over a greater area. And actually Thierry Basisson in New Jersey, he's actually going to be doing a vision guided sprayer trial through the IR4 program in blueberries. And I'm doing the vision guided sprayer trial here in New York in grapes to look at new herbicide active ingredients that are going to now be safer for use because of the way we're using them.
Great, thank you. All right, we have the second, this is the second question of the 3 poll questions, and it looks like people are answering that.
Nope, I'm going to give him 10 more seconds on this one.
Okay, thanks, Natasha.
All right. Just so you all can see the results of that real quick. And then the third poll. So you'll have about a minute and a half to answer this one as well.
And Lynn, there's another question from a grower in the chat box. Would you mind answering that, just chatting that answer back to everyone?
Sure, I will get on that. I'm sorry, my technician texted me, so I wasn't looking at the.
No problem. It just came. I'm just drawing your attention to it.
I'll look for it.
So we'll leave it up for another 30 seconds or so. Just a reminder, if you are getting DEC credits for the session, you do have to answer the questions.
And I'm going to go ahead and introduce Esther Kibby because we've really eaten into her. Esther, you don't have to try to smush it all into that sort of time. We can go over a few minutes. I apologize to everybody, but we had really a lot of dense topics, and so I appreciate everybody hanging in there. But Esther Kibby is the Cornell Cooperative Extension Harvest New York program, which she is responsible for berry crop extension and applied research in the western part of the state. And I'm really happy to have her on board. It's been tremendous to have a colleague out in the other part of the state. So, and she's going to talk today about diseases. And as soon as we are done with the polling, we can move to that presentation.
Yep, we're all done polling.
All right, thanks, Natasha.
All right, thank you, Laura. And it's great to be here with you all today. Before I jump in, I will make a comment. This is going to be focused on blueberry pest problems. So there's certainly many other problems. I'm glad we had Lynn because I know weeds are a big one. But I won't be talking about weeds. I won't be talking about diseases. I'm going to be focused exclusively on pest issues that I've been seeing recently out in grower fields and just the things I've been advising on the most. So the first is spotted wing Drosophila, and I'm not going to talk about this one. I'm going to move pretty quickly through this because you had just such an excellent presentation from Greg already this morning, and I think you're all pretty familiar with this one, but you can't talk about pests and blueberries without mentioning it. And you know, again, I think Greg did a great job covering the current and more innovative steps towards managing SWD. So I'll just keep moving along here. The next pest that I've been seeing more and more of is this one, which is blueberry stem gall wasp. And this one has just been cropping up as a more significant issue in many blueberry fields. And it seems that just based on some research and observations, that the reason we're seeing more of this is that there have historically been a couple of parasitic wasps that would go after these galls and parasitize the larvae inside the galls during the summer. And that's when we're doing all of our SWD sprays. So that seems to be kind of that reason why we're just seeing so much more of this pest when it was historically just kind of a minor issue that we weren't seeing very much of. And it's also very much a variety linked pest. And so There are certain varieties that show a lot more of this, such as Jersey, Northland, and Blue Jay. And in Michigan, this has become so bad, and they have more of these varieties that they've been taking out entire blocks just because they've got no other way to manage it effectively. And what we know about this pest is that the wasps are emerging from the galls during bloom, and then they're quickly mating and then relaying eggs. And that is a challenge for control because we're really trying to avoid using insecticides during bloom. So how do we manage this? The first, of course, is pruning, and this works if you have a low level of infestation. You can get out there and remove those galls during the winter pruning. Or you can choose varieties that are not as susceptible to this. And I know that the Michigan blueberry grower is doing some projects and making it a focus of his breeding efforts to have any new varieties or cultivars be resistant to this pest. And then in terms of insecticides, you can spray for this. We really encourage, and I think by the label, you have to wait until after bloom or when the bees are removed to go in and spray for this pest. The only product that we have labeled in New York is Vertiprin, and it has a 2E label. And the recommendation is 2 applications 10 days apart. So 1 right when the bees are removed and then 10 days later. And you want to use a penetrating adjuvant with that, because if you want it to get into the gall to take out those larvae and eggs that are developing, you really need to make sure that it can get into the tissue. And the other thing to think of is that some of the post-bloom sprays, if you're controlling for cranberry or cherry fruitworm, that this may also have, even though the focus is those pests, it may also help control this one. And then speaking of cranberry and cherry fruitworm, these are ones that I'm seeing a fair bit of, more so in organic systems and maybe some of the lower spray blueberry plantings, but it is certainly a concern. And one of the things that you look for is berries that are starting to turn a little early. And it's easy when you see this kind of shriveling berry to think, oh, it must be some mummy berry maybe. And then you remove that berry and you see this is a cranberry fruitworm. And the way you can tell them apart is the cranberry fruitworms have the webbing and they are feeding more from the outside of the berry. And so they may like web this cluster together like you see here. And then the cherry fruitworm tends to be more inside the berry. So you might see a very small scar like this one. And then you open that up and you'll see that little reddish worm or larva that's in there. And then the frass or excrement from that insect larva are inside. So you don't, with the cranberry fruitworm, you're going to see that outside and kind of in with that webbing. And then with the cherry fruitworm, always inside the fruit. But the same kind of thing, you're going to see a berry that's starting to turn color a little earlier than the rest of the fruit. So one personal note is be very careful about thinking, ooh, these ones are starting to turn and trying to pick those. But in fact, that is a management technique. If you're starting to see these berries color prematurely, if you don't have a super heavy infestation, you can just go out there and remove those clusters or remove those individual berries that have turned color early, and it kind of signals that you've got an issue there. And then just remove those by hand and then remove them from the field and destroy them. But that can help reduce your infestation levels if it's not too severe. And the life cycles of these fruitworms are very similar. So I'm kind of treating them together to kind of see them at the same time as well in the field. The larvae overwinter in cocoons on the ground. And then the moths emerge around bloom time and they lay their eggs in or on the berries. And then, as I said and showed you, the cherry fruitworms feed inside and then inside the berries, and the cranberry fruitworms tend to web those berries together and then have external feeding. And then in terms of management, again, as I said, you can remove the damaged fruit, or you can use insecticides, and those are applied at petal fall. So again, sort of a post-bloom application, and then maybe 10 days later. The products labeled for excellent control are Exirel, 7, Asana, Acetyl, Delegate, Ulticore. And these will also help with some of your other pests that you might be having around this time that maybe are slightly less common, of leaf hoppers or leaf rollers or gall wasps. One thing to always be thinking about is some of these are the same products you might be using later for SWD, and those have season-long limitations on applications. So be sure you're counting those applications in, or sort of choosing things that aren't going to mess up your later SWD management.
Esther, do you mind if I interrupt right now? We've just got a couple of questions about organic control, and one was, are there any other organic spray options for these pests that would be useful? And do you have any suggestions on cranberry fruitworm scouting for organic spray timing?
So the challenge again is because they're emerging around bloom, in terms of if you're seeing it, and I think it's more of like if you see it one year, you want to control for it the next year. I'm not really sure how to scout for it during bloom, because then you start seeing it showing up later. And I know the product that I was seeing listed was BT, for managed organic control on this one, because they are worms, or in that category.
And I know we've used a lot of the pheromone traps, so that is a good way to kind of give you a little bit of an idea of how heavy the pressure is. The problem is that timing is it's really tight, so you have to be on your game to use them effectively, in my experience.
Yes, yeah, and that's a good point. And one I didn't mention is that there are absolutely pheromone traps that will work for these to check if you've got some flying out there. So yeah, thank you for adding that, Laura. The next pest that was very bad this season and this past year, and then maybe in many seasons can be very challenging, is Japanese beetles. I'm sure you're all very familiar with this pest on multiple crops. And these are challenging, of course, because they're good flyers, they have multiple hosts, and they are just ubiquitous in our state and region. And then the feeding can be very damaging, and they just kind of make a mess, and they're unsightly and just generally unpleasant. And kind of the most effective way to manage them in a localized setting and sort of deal with the life cycle on your own property is to treat the soil or the sod with nematodes or milky spore that can help control the life cycle locally. Milky spore will work, but it's something you're going to have, it's expensive, and you have to continue to use it because it tends not to last through the winters very well. So in other parts of the country, you can put milky spore down and it just becomes endemic in your soils, and it really has this long-term management effect. And here in New York, we've found that you really have to put it out every couple of years because it tends to dissipate and not survive. But the nematodes are, again, expensive but effective. And there are multiple types that show good efficacy against Japanese beetle larvae in the soil. And then you can use insecticides against the adult beetles. There's many things labeled. Imidan, Brigade, and Danitol have been listed as having excellent control. And again, we're sort of back in that issue of, how many applications are you allowed of different products per season, and working that in with your SWD management. And some of it, it's really disheartening to see those masses of Japanese beetles out on your bushes. Though, and there's some question about how much economic injury is it causing? And sometimes it absolutely is, and sometimes it may be more unsightly, and then it just, they do their thing and they're gone, and you have the rest of your season and you realize, well, maybe it wasn't that much actual damage. But again, that depends on the timing of your crop. If you have earlier blueberries, I think it tends to be a worse issue that you're seeing. So I think also weigh that before you go out there with a chemical treatment. And then this one, Plum Curculio, I wanted to mention, I've seen it again, particularly in some organic fields where there's just not much insecticide around. And so this is what we generally consider a minor pest in blueberries, but it does show up. It's really a bigger pest in tree fruit. So some of this is probably location and how close to untreated tree fruit orchards or wild hosts of wild apples or whatever. And really the thing to be looking for is this characteristic crescent-shaped scar, and then this affected fruit will drop generally before the rest of the crop ripens. But if you have an early variety, sometimes it hasn't dropped, and then you're ending up with infested fruit. And you can, again, just tell by seeing the fruit starting to turn blue a little earlier than the rest of it, the rest of your crop. And again, probably it's not a major pest in most conventionally managed fields. And, for many operators, you could probably just remove or destroy the berries before they drop off and kind of break that life cycle. But if you need to, there are insecticides that are labeled for this pest in blueberries. And there are others that are rated as good control. And again, that would be sort of a post-bloom spray to take care of that. All right, so I moved through those pretty quickly, but hopefully we got us back on time, and I'd be happy to take any questions.
Thanks, Esther. And I want to apologize to you and the growers. For some reason, I still had the disease topic in my head, and I apologize. I do remember that you said what you were going to do and you did what you were going to do, and I just did not make that mental switch. So I apologize to everybody if you were looking forward to specifically disease information. That was my problem, and now we owe you. We'll have to have some kind of a disease workshop if that's of interest. Does anybody have any other? Lynn's been posting some very good information about HRAC classes. That's very helpful. And I did put, there was a question about the scouting and using traps. And so there was a nice little article in one of the New York Fruit Quarterlies about how to use those pheromone traps. I think there's also another fact sheet that Molly did, Molly Shaw, quite a long time ago actually, but still very good information. And I can try to find that here and post that. And does anybody else have any questions or comments? I was trying to tell you folks about one organic grower that had a very heavy cranberry fruitworm infestation and did successfully remove the clusters. It was pretty successful, but it took a ton of labor. And then there is another post directly to me about the fact that Entrust and Natural Light are also labeled for cranberry fruitworm, so they might help with organic control. And Jay has a question about new information on overwintering SWD, and this might be for either Esther or Greg. Any new information on that front?
Let Greg take that one.
Right. Well, it's still not 100% sure where and how they overwinter. We don't think a lot of them survive, but they clearly overwinter in pretty cold places. I'm sure they're overwintering in New York. what we see is a pattern, and we've done experiments testing it, though it's hard to test. We see a pattern is if we have a really cold, kind of rough winter, populations tend to show up later and get to lower numbers. They see this in Europe as well. So that kind of confirms to me that, yeah, given the right conditions, they survive. If it's a harsh winter, not very many of them survive. If it's a mild winter, more of them survive. And we hope someday maybe we'll be able to use that to give us a little bit of a early warning of, you know, is this going to be a bad year or a good year for spotted wing pressure? We're doing an experiment actually this winter. We're just running it right now. One of the things we think prevents them from successfully overwintering in our area is we have a lot of freeze thaws. And so we're testing the hypothesis that when snow melts and then refreezes, that's actually really hard for these things. If it just stayed snowy, they might actually do better over at least some of the winter. They do need to eat if it does warm up at all. That's another problem for spotted wing. They actually have to eat or they'll starve to death. So, you know, I don't think it's an easy thing for them to do to make it. But, you know, I remember when I first gave a talk in 2012, I'm going, well, maybe our cold winters will be of benefit to us and really helping us get rid of this thing. And although I think it helps us, it clearly is not harsh enough. This is an area they can do okay. And we're still trying to understand, maybe we get some migration from the south. We're still not clear on that. There's a lot of gene exchange between the north and the south in the eastern United States. And whether that's also contributing to our populations, I think in some years it probably is, but I'm pretty sure they're overwintering as well.
All right, I wonder if that bodes poorly. We've had relatively consistent snow cover where I am, but that's interesting. Thanks,
Yeah,
Greg.
well, maybe when it melts this next week, and they'll drown and freeze. Although they don't drown that well, but yeah.
Can only hope.
Yeah,
Sounds
exactly.
like an excellent end for them.
Yeah, if.
And I just put in one more just to draw your attention. If you're still looking for DEC credits, or if you're specifically if you're a tree fruit grower, there's a bunch of great workshops that are going to be offered during March. They're free, and we have been able to work out reciprocity with DEC for the credits. So if you still need another credit or two, if you're looking for specific information on bitter pit, I think there's a stone fruit webinar. Anyway, that is there if you click that link. But I also want to make sure to draw your attention to the chat box, and Lynn put up Geneva's looking for a manager. So that's great if you're looking for a job. I want to thank everybody. Thank the speakers, Esther, Greg, and Lynn, for being here. Thank our technical support, Chelsea, Natasha, and Andy. And oops, I have one more question. Is there any new SWT trap material that is better to use than what is currently used? As far as I'm assuming that this is when you say trap material, you're talking about a drowning trap?
I'm guessing they're referring to monitoring traps. Is that?
Yeah, I think so.
So there is something that Julia, Carol, I think Esther, maybe Esther, you involved with that as well? A test of a dry trap, a red dry trap that I believe not Sutera, but Tracy is making, the company Tracy. You have to use it for looking at males. And I think in blueberries, it actually might be quite helpful. There's an attractant with it, and it's a sticky card, and you look for males. And our research is that males are a pretty good indicator of impending infestation risk. It's not so much true for raspberries for various reasons we don't fully understand. We tested a version of this trap earlier on. We weren't very impressed with it, but I think that they've redesigned it, and a lot of my colleagues are pretty high on it because it's something that it's pretty easy for growers to use. They don't have to get out the dissecting scope. They don't have to change drowning solutions. So I know Julie, maybe Esther wants to talk about this a bit more, but the whole region is going to be testing it. And then in our work, I think I'm also going to include it in ours and compare it to our drowning traps again, because I want to look at this new version.
Yeah, Julie had submitted a grant request for that. Unfortunately, it wasn't funded for this year. So
Okay.
I'm not sure kind of the status of that project now. Certainly there's products out there, and I think it's definitely, as you say, Greg, very promising. And I hope we can get to the point where we're doing more testing and validating of that system. And, you know, even if it means looking at what folks are doing in other regions. But yeah, unfortunately, that one wasn't funded for our local region.
Okay, that's too bad.
Yeah, we might be able to get the information about the traps out to people so they can anecdotally try them themselves, and that might be very useful for growers, but I don't know if we're going to be able to do quite the protocol that Julia had suggested.
Yeah, I do think we'll include them with our other traps and fruit infestation at a couple farms that we're going to work in more intensively, part of that SCRI grant, because that's one of our things that we think are ready to implement is improved monitoring. The infestation part's really important. We just haven't, I don't think, figured out a good way to make it easy for growers to use. But I do think if they can or will use it, that might provide them the best information. But maybe for blueberries in particular, that male identification might be just as useful. So yeah, we'll get a little bit of data, and I agree that our colleagues are looking at it in Rutgers at a number of farms. Maybe Anna might still be doing something in New Hampshire. I don't know. So we'll keep you posted on that.
Right, great. And this recording, this workshop has been recorded. We'll post that recording on the Teachable. Natasha had typed in information for how to get to the Teachable so that you can listen to the recording if you wanted to pick something up that you missed during the actual presentation. So thank you, all of you, for hanging in there. It's been a long winter. It's amazing to me how these Zoom things, as handy as they are, they do kind of sap your strength after a while. And I think just being with one another is kind of invigorating, but hopefully we'll be together sometime in the future.
Soon.
Yes. All right. Thank you all. Take care.